Hello everyone, this is Sajal Murshid Sajib, a PhD fellow at Tuskegee University, USA. Today I'm going to discuss on KAISO-linked genomic disparities in breast cancer of African American patients. First of all, I would love to discuss about the cancer development and its process of being metastasized. Uh, the development of cancer initiates when a single um, mutated cell begins to proliferate abnormally. Later, as an example, let's see the breast cancer progression from the normal breast epithelium, where the cells are well organized. Later on, the cell proliferation happens abnormally and creating primary hyperplasia, then carcinoma in situ. That is zero. We can tell that zero stage of cancer, and that's curable, obviously, and that later on it become invasive carcinoma. The figure shows in the right that epigenetic regulation and tumor microenvironment and immune interaction drive breast cancer progression, stemness, and metastasis. Process begins with the blocking of tumor suppressor gene expression, then epigenetic changes like DNA methylation and the histone modification plays a role in decreasing of E-cadherin expression, that is mainly responsible for cell adhesion, increase EMT metastasis, and tumor microenvironment secretes tumorogenic factors, promote tumor growth, and enhance inflammation. Immune cells are recruited by reprogrammed. Tumor microenvironment plus epigenetic changes activate cancer stem cells. After dissemination, cancer cell can revert to the mesenchymal epithelial transition that forms secondary tumor in metastasized region after travel to the region. Let's check some statistics for cancer from global perspective and US perspective. In these bar charts, we can see that the lung cancer is the highest incidence happen in worldwide, and the mortality rate also highest in worldwide. But in case of US, the prostate cancer is the highest in term of incidence, and the lung cancer is highest in the within the US in terms of mortality, and the breast cancer is the second highest in terms of incidence in both US and globally, and the fourth highest in terms of mortality percentages in both globally and the USA. And that is one of the reason of selecting breast cancer for my research perspective. Let's go a bit deeper with the breast cancer subtypes and the treatment outcomes. There have several ways of classifying the breast cancer subtypes, like classifying according to the histological subtype or molecular subtype, or can be TNBC subtypes and genetic subtypes of TNBC. But the most prominent classification is PAM50, that is actually based on the prediction analysis of microarray using 50 gene signatures. Here in this chart, you can see the different type of PAM50 subtypes called luminal A, luminal B, hard to negative, and basal-like TNBC. Actually, this basal-like TNBC is also can be subclassified like basal-like 1, basal-like 2, mesenchymal luminal androgen receptor TNBC, and finally the most aggressive one, the QNBC, that is also not only uh, um, uh, absent with androgen receptor, progesterone receptor, and human epidermal growth factor receptor. It's also uh, absent of androgen receptor. So, in terms of aggressiveness and treatment response, you can see here I used the green one for like low risk and less aggressive. The yellow one is moderate risk and um, moderate treatment response, and red one is for poor treatment outcome and high risk of cancer, aggressive cancer, and the double red that is indicate the extreme risk in terms of aggressive breast cancer. So here we can see the aggressiveness increases according to from luminal A to the basal like and TNBC, and in TNBC it increases from basal like 1 to the QNBC. Uh, let's check some breast cancer disparities. Those played a vital role in selecting my breast cancer research. Incidence rate low but mortality rate high in African American. That makes sense. Aggressive basal-like or TNBC subtype breast cancer form is significantly high in African American, and they mostly have the um, androgen receptor negative tumor subtypes higher amount, and also in Q1BC they shows higher basal like 1 and basal like 2 subtype aggressive subtypes in African American patients. Let's check some molecular level disparity in the most aggressive breast cancer type QNBC, that means quadruple negative breast cancer. That is a special type of triple negative breast cancer that lack of androgen receptor. Usually, the TNBC has the lack of estrogen receptor, progesterone receptor, and human epidermal growth factor receptor. Here we can see that the African American black have been or black QNBC have been shown to exhibit significant upregulated level of T cell marker antigen, CD4, and immune checkpoints and genes like PDL1, PD1, and CTL4. These markers should be good for immune response and chemotherapeutic response, but they have a very interesting point, like the excessive and persistently high amount of T cell markers indicate immune exhaustion that is characterized by increased expression of immune checkpoints like PDL1, PD1, these things. And actually, this means that the immune system is actively trying to fight against the cancer and suppress the cancer cells, but the tumor microenvironment is not helping that. Uh, In summary of the breast cancer disparity, we can say that African American women experiences higher mortality rates, more aggressive breast cancer, and higher incidence of triple negative breast cancer, and also the quadruple negative breast cancer. The molecular level mechanism is really still unclear, and the major question, what regulatory mechanisms actually driving this aggressive breast cancer in African American patients. We are suspecting that some epigenetic regulation and transcription factors might play a vital role. Why we are suspecting these, we're gonna make it clear in next. Let's see how epigenetic mechanism works. Epigenetic means something that is living outside the DNA and regulating the expression of genes. Those are inside the DNA. These are external factors. Those are playing vital role in health outcomes like cancer, autoimmune diseases, mental health disorder, and diabetes. This epigenetic influence starts from the early stage of development, like uterine development or early childhood development, and influenced by environmental factors, chemicals, drugs, pharmaceutical molecules, aging, and diet. This epigenetic regulation may happen in two ways, like DNA methylation or histone modification. DNA methylation process, methyl group can be tagged with DNA and a specific place of DNA and activate or repress a specific gene. While the histone modification includes the modification of histone binding with the DNA by epigenetic factors. The histone binding with the DNA that determines the compactness of DNA that actually uh, or wrapping of the DNA around the histones and the availability of the genes in the DNA to be activated. Here we can see that the transcription, how transcription factors works and how it stops the transcription process, like the binding of DNMT, like DNA methyltransferase, methyl binding protein, histone deacetylase. These things binds with the DNA and like suppress the transcription by uh, make the specific gene more or less accessible by the RNA polymerase. Then on the other hand, like histone acetyltransferase can make it more accessible to go through the transcription process. Let's discuss some previous uh, experiment on uh, important transcription factor KAISO that plays a vital role in epigenetic regulation and the breast cancer disparity in African American patients. Here we can see that the research shows that less survival rate for the patients who are highly positive with the nuclear KAISO, and this continues to show in terms of like metastasis level. Like higher the metastasis level, higher the nuclear KAISO expression, and at the same time lower the E-cadherin expression. And also it shows interestingly, it shows that the African American aggressive breast cancer shows most highest amount of nuclear KAISO expression rather than the European American aggressive TNBC, say. Um, so we can say that transcription factor KAISO might be a cause of molecular disparity and cancer aggressiveness in African American patients. Here we can see in the left that the different Nigerian, Barbadian, African American, and Caucasian race is for transcription factor KAISO expression. The red one, it shows the nuclear KAISO expression, and the blue arrows indicate the cytoplasmic KAISO staining. We can see the nuclear KAISO high AH score is much higher in African American than a Caucasian American, and that's a significant thing. And also, we can see on previous study, like KAISO is highly nuclear KAISO highly is higher in primary tumor than normal tumor, and in metastatic tumor than primary tumor. Also, the interesting thing is, like you can see in African American primary tumor and metastatic tumor is shows higher nuclear KAISO expression than a Caucasian Caucasian primary tumor and metastatic tumor. Here in the back, left side, we can see that the KAISO expression in normal cell, adjacent normal cell, then oil differentiated tumor, where upregulated KAISO is expression within the cytoplasm with nuclear positivity. And then we can also see that the moderately and poorly differentiated tumor showed intense cytoplasmic and nuclear positivity in D and E. And finally, the lymph node metastasis demonstrated the low cytoplasmic KAISO and intense nuclear KAISO. So now let's see how KAISO works for this aggressiveness of cancer, and how it regulates the metastatic things, or or cancer related things. First of all, what we can say that KAISO is a BTB positive transcription factor. For it has BTB pause, protein-protein interaction domain, and 3 DNA binding protein DNA binding domain for zinc finger domain. And though KAISO negatively regulates expression of many genes uh, in a methylation dependent manner, but also positively regulates through the KAISO specific binding sites. Like it actually inhibits the E-cadherin expression, tumor microRNA 31 family, microRNA 200 family, and that is actually how it make cells cancer cell more metastatic. And also through the KBS side, it activates BRCA1 like genes that actually, and inhibits cyclin D1. So that actually increases the tumor proliferation. And also in simulation also plays a vital role in case of KAISO specific binding site dependent gene activation and depression. Like while we can see that the TRIM25 that is activated when KBS is simulated, and is inhibited by KAISO specific inhibited by KAISO when it's not simulated. This TRIM25 actually helps. It's the ubiquitin protein, and that helps to degrade the p53 apoptotic gene. And also in case of wild type p53, KBS actually activates the pro-apoptotic genes to make it more aggressive. And it also interacts with the TGF-beta signaling and activating the SMAD1, 2, 3, 4, and transferring those to the inside the nucleus, and activating of KAISO, JAB1, JAB2, and decreasing the E-cadherin expression, that in terms actually make it more aggressive and metastatic cancer. And also, like KAISO expression represses E-cadherin expression through depression of microRNA 200, and possibly via sequestration of P120 catenin in the nucleus. Phosphorylated P120 catenin binds KAISO, which leads to its sequestration in the nucleus and E-cadherin degradation. So all this way actually KAISO making more aggressive breast cancer, and also helping in the disparity in breast cancer of upper income patients. Okay, now let's discuss a more recent ATAC study on KAISO that shows something different than our previous discussion. In this ATAC study, KAISO is showing in the open chromatic region of African American breast cancer cell lines only, and also shows the specific cancer signature genes expressed in these African American aggressive cell lines like HCC 1806 and 468. And this says something different, that like previously we discussed that actually the KAISO shows the disparity and the aggressiveness of cancer through a methylation dependent gene depression. But here open chromatin KAISO is in the open chromatin region that gives us some other view. Created another horizon. Um, from this study, our preliminary observation is KAISO is understood in open chromatin region of African American breast cancer, aggressive breast cancer cell lines, and this is unexpected for methyl binding transcriptional repressor KAISO. So our suggestion is alternative binding mechanism may exist, possible explanation that KAISO binding to the KBS motifs, and protein interaction mediated recurrence of other cancer mediators. So our primary hypothesis is like KAISO contributes to the aggressive breast cancer in African American patients through non-canonical binding mechanism, and that may be the KBS binding motif, and also the other regulatory interaction with the other regulatory proteins and regulation of transcription in open chromatin region. So we went through all the multi-omic approaches, all possible multi-omic approaches to integrate the pipeline, like cut and run for to detect the KAISO binding pattern with the DNA, and attack sec overlap to check if that binding happens in the with um, other genes, major genes in open chromatin region, and also checking the DNA methylation pattern, like that are regulating or inhibiting or giving activating other genes, and also the changes of gene expression through the RNA-seq, and also the protein interaction, like co-regulator proteins through the RIME assay. This is one of the our research outcome that presented on Americanization of Cancer Research uh, conference, disparity conference in last September, titled as KAISO defines a distinct chromatin and protein interaction network in breast cancer, implications for precision therapy. So, in summary, we can say that ZBTB33 gene, known as KAISO protein, and this is a is mainly a methyl DNA binding protein, also a transcription factor. It recognizes two DNA motifs, like one is methyl binding motif CGCG site, and another one is KAISO binding sequence, that is KVS, and mainly function as transcriptional repressor, chromatin regulator, and implicated in tumor progression, epithelial mesenchymal transition (EMT), and Wnt signaling. The major key knowledge gap is KAISO binding behavior in breast cancer disparity is still poorly understood. So, in summary, we can say that our target is to compare the metastatic cell lines of African American and European American, also metastatic with non-metastatic cell lines, to check the uh, overall system regulation for KAISO, and these are this would be done with the major analysis techniques like cut and run, by which we can check the uh, KAISO binding site, a specific KAISO binding site in DNA, and that is how we can check whether it's binding mostly binding with the CPG specific site or KAISO binding specific site, or we can comply that with the attack sec data, and also we can check the dependence of methylation for KAISO aggressiveness, like whether this KAISO aggressiveness is methylation dependent or in methylation independent. And also we can check the overall genomic expression changes in these aggressive breast cancer cell lines in African American and European American through RNA-seq analysis. And also we have a plan to check the KAISO interaction partners, like the other proteins. Those are acting as co-regulator or co-activator for KAISO, and changing the activity of uh, cancer aggressiveness. Thank you for your patience and listening my lecture. Have a good day.